In this tutorial, I will be showing how to do a data analysis to examine a gene of interest using a depositor-processed expression data, focusing on the genotype, which spans across all datasets, and the results will be portrayed in a dot plot with the x-axis showing the value, the y-axis showing the group, and the results being prioritized by statistical significance, specifically p-values, and colored by selected features. So as we can see here, we are going to switch from the data to analysis portion. And under analysis options, we're going to have the pre-selected examined by gene of interest selected. And since we are doing a depositor process data, that's also going to be automatically selected. But if not, you can go ahead and select that. And this is going to show the gene expression data specification. For my gene of interest today, I'm going to search EZH2, and for our comparisons, there's 3 types of comparisons options. So as I showed in the flowchart, we're going to focus on genotype/treatment, which spans across all data sets, while the second and third focus on subsets of pre-selected data sets. So selecting genotype treatment, I'm able to move on to the filter input samples. So this is a reactive filter, which means it's used to refine input data and review the selection in the tree below. And so something to keep in mind is that the filters are dependent on prior filters. If I search a bulk tissue for my sample, and then for my primary metastasis status, I search and click lung, that means all the lung results I get are going to be bulk tissue. So it's going to be dependent of the filters that I put before it. And so this makes everything very dynamic. And I can go ahead and also input sample, primary metastasis status, the histology, the sex, the strain, and the genotype. So particularly for filters such as sex and strain, they're included to help refine searches. But something to keep in mind is that missing or imbalanced metadata in some of the datasets may limit the utility of these filters for detailed subgroup analysis. And as we can see here below in the tree, we have all the samples, and all of these are called datasets. So dataset names include descriptions of common sample features. So we can see GSE 10954 treatment, and we can see none in bulk tissue. And then if I click the little plus sign, I'm able to see all of the sample names that display unique features for each entry. So once I do all my filters, in this case I have just bulk tissue, and once again my gene is just EZH2, I can go ahead and click submit, and it may take up to 30 seconds to generate the results. But just be patient with it, and once the results are generated, we're able to see the studies ranked by significance with the lowest p-value from 1Y ANOVA shown first. And there are a lot of studies shown, as I said, as this goes across all data sets. And the genotype comparison will generate a plot displaying results from the selected data sets and samples. And that is it for this tutorial. For the next tutorial, we will be showing how to examine a gene of interest using a depositor process expression data, but we'll be comparing by primary metastasis status.